hopefully we'll have more success today than we did yesterday. So chapter 11 is about the evolution of populations, microevolution. Now we've been talking all along about how we like variation. Variation is a good thing. It allows more organisms to survive. Because if the environment changes and there's variation, yes, some will die off, but some will be resourceful, as you were yesterday as little finches, and be able to get the resources, and then they'll carry those traits on to their offspring. Now, I used the term earlier in the week, gene pool, and so we're going to be talking about variations in populations in their gene pool. So we need to define that term. A gene pool is all of the alleles that are present in a population. All of the alleles that are present in a population. So what we're going to be looking at today is how that gene pool changes, factors that affect the gene pool. But we'll start with, where does that basic genetic variation come from? There are two sources of genetic variation. Mutations. So changes in the, if we have a mutation, that's going to change the alleles that are present. And recombination. So this goes back to meiosis, producing those unique gametes that must come together in sexual reproduction. So we're recombining the genes, putting new alleles together. Now, when we take a look at natural selection in populations, we're looking at the distribution of alleles. And really what we're concerned with are the phenotypes. So we're looking at how are the alleles expressed? What are we actually seeing in these organisms? So there are some patterns that we can see. We can see a normal distribution. Now, we have such a small population, it's hard for us to kind of take a look at our distribution patterns because we're such a small population. So this is what normal distribution looks like. So the mid-range is the most popular. So we could do something like height. Okay? So human height. And if we got the whole population of Duchenne, because that would give us a little bit bigger number than what we've got just in our class, since our class is only 12, okay, that's a pretty small population. So we have the whole population of Duchenne, and we go around, we do a survey, how tall are you? And we count up the number of people that are each height. The majority are going to be in the middle. So we'll have some middle ground, like maybe, I don't know, 5'6" or something like that. And that's what the majority are going to be. And then as we get further away from that, there are fewer people that are down here in the 4'10" range, let's say, and there are fewer people that are down here in, let's say, the 6-foot range. But the majority are in the middle. So that's normal distribution. And so for these others, we're going to compare them back to normal distribution. So directional selection is when one extreme phenotype is favored. I have some laser discs from an old biology book, and they have an excellent example of directional selection on them. And maybe I'll dig them out next week and see if I can get them working. But their example is with moths during the Industrial Revolution. So the moths, they call them peppered moths, are kind of grayish in color, and they blend in really well with tree bark. And then there are these same kind of moths, there are black ones. Well, they don't blend in with the tree bark, they stand out. And so they're more likely to get eaten by the birds, and the peppered ones, the gray ones, are less likely. So the peppered is favored. And so what we see before the Industrial Revolution is that the population, the peppered moths were favored, so there was a higher population of the peppered moths and a lower population of the black moths. So this dotted line is our normal distribution, but the population after selection is higher towards one extreme phenotype. The example in your book, which you'll read about in your assignment, is about antibiotic-resistant bacteria. I'm very excited. This morning in my email, I got a lab about antibiotic resistance in bacteria that we can do next week. Yay. I have to get the stuff for it, though. That's the only thing. All right, stabilizing selection. Stabilizing selection is when an intermediate phenotype is favored. So your book gives the example of gall flies. Gall flies are tasty for wasps and for woodpeckers. Now, if you think about the size of wasps versus woodpeckers, woodpeckers are relatively big birds. I mean, not relative to like a hawk or something like that, but they are pretty big birds usually. Okay, so the wasps like to eat the little gall flies because wasps are little. The woodpeckers like to eat the big gall flies. And so that intermediate size, that middle-sized gall fly, that's the phenotype that's favored if you're a gall fly, because you're not going to get eaten by a wasp, you're not going to get eaten by a woodpecker. And so the population of medium-sized gall flies is highest.
So then, what's the difference between like normal distribution and stabilizing selection? Because I mean, if they're both like in the middle.
They're both in the middle, but notice how we have a greater number of that are here, because this is the normal distribution, is the dotted line. So there's still a greater number of extremes. There are fewer extremes in stabilizing selection. Disruptive selection is when the two extreme phenotypes are favored. So the intermediate is not favored. It's where the two extreme phenotypes are favored. An example of this are lazuli buntings. They're a bird. And lazuli buntings are either bright blue or brown. And we're concerned with the males because, of course, the females have the mate choice. And females prefer the really bright blue. That's what they really like. So you would think that it would lean in that favor, but the bright blue males pick on the kind of bluish brown males, the younger ones that haven't fully come into their blueness. And so the brown males that haven't even started to turn blue yet are able to get mates because the bright blue ones are picking on the ones that are starting to turn blue and not allowing them to mate. So the bright blue ones are mating and the brown ones are mating. And so the two extremes are favored. But if you're somewhere in the middle, you're going to get picked on by the bright blue ones and you're not going to be able to mate. Kind of a weird scenario. But that's what disruptive selection is. So some other mechanisms of evolution, and once again, we're still talking microevolution. We're talking about evolution in populations. Genetic drift. Genetic drift is changes in allele frequencies in a population due to chance. Sorry, I should have gone down. Sorry, this will be under general. Genetic drift causes a loss of diversity in a population. So there are a couple of ways that this can happen. These are the most common ways, I guess, it can happen. So one way is, where'd it go? Okay, that should be an A. The bottleneck effect. The bottleneck effect occurs after something happens, an event happens that greatly reduces the size of the population. this is going to be a little bit tough since we're such a small population. Okay. So we're reducing the size of the population, greatly reducing it. So it could be something like, it could be like a natural disaster, fire, something like that. Something that wipes out a huge amount of the population. And so that's leaving us with a smaller gene pool, which means less diversity. So how many of you can roll your tongue? I can't do it, so I can't. Is there anybody else who can besides me? Can you do it? Okay. Nope. Can you do it? Okay, very good. So there's like 3 of us that can't roll our tongues. Okay, very good. So this is our population, and like I said, it's kind of goofy because we've got a very small population. So we're just looking at the tongue rolling allele. It is a dominant allele. And so if something wipes out, so we've got our wonderful snowstorm going on out here. Let's say that our storm wipes out this part of our population, everybody from Mira over this way. So now this is all that's left of our population. All of you can roll your tongues. We've wiped out all the people who can't roll their tongues. And so your alleles, your gene pool has changed dramatically. So the reason why they call this a bottleneck effect, it's as if you had your alleles in a bottle. So you put like different colored marbles in a bottle to represent your alleles. Let's say you have 3 different colors of marbles in there, and you're pouring them out. When you pour them out, because the bottleneck is so narrow, you don't get very many out, and so that changes your gene pool. You don't have all the alleles that you used to have. You don't have the same frequency of alleles that you used to have. All right, the founder effect. Is when a small number of individuals colonize a new area. So you're starting your own gene pool. So for instance, when I give you guys time to work on your homework and there is a small group that always goes back between the 2 lab tables back there, okay? You're colonizing a new area. You have your own gene pool. You're starting fresh from that small gene pool. Another mechanism of evolution, gene flow. Which is the movement of alleles from one population to another. So we're experiencing this today because 2 members of our regular population have moved out, at least temporarily, and we have added a new member to our population. So we're experiencing gene flow. Our gene pool is different today than it was, let's say, yesterday even. We could talk about it in terms of like our city, the population of our city. When people move to our city, they're changing our gene pool. When they move out of our city, they're changing our gene pool. Sexual selection can also have an effect on evolution. And we've already talked about this a little bit. So sexual selection occurs when certain traits increase mating success. Now remember, okay, so males produce sperm continuously. And this is true of all organisms. Once they reach their reproductive age, they produce sperm continuously. Females, however, are more selective and do not produce eggs continuously. And so the females get to choose the male. Mate choice is all on her. So there's going to be competition. This is a resource. A mate is a resource. There is intrasexual selection or competition. And this is competition between, usually, between the males. So my husband and I used to live in this apartment complex, and there was a big courtyard and lots of big tall trees. And cardinals mate for life. And we had a pair of cardinals that were living there, and the way that he woos her is by singing to her. Now, she also likes it if he's very bright red. That's a sign that he's a healthy male. But the song is how he kind of woos her in. And so this male cardinal would sit up on the top of the tree and call to her. And my husband, being the snot that he is, could mimic that call and would do it. Tick that cardinal off. He had competition for his woman. He did not like that at all. That would be kind of an example of intrasexual selection. Okay. Intersexual selection is when males display certain traits to females to try and prove that they're the best mate for her. So, like when peacocks, when they fan their tails out, my beautiful tail. There are birds that build little bowers where they collect shiny objects to attract the female. And they're very particular about their shiny objects. We'll watch a video later in the year where this scientist is there. He's a naturalist. He's observing nature. And he goes into one of these little bowers and he moves the stuff around, and it really ticks off the bird because he wants his bower perfect to attract the females. Or one of my favorites is this little bird, and he's black, and he does a fan dance to attract her. So he's in that same video. We'll see him too. He's pretty funny. All right. And I believe, are there any questions? I think that wraps it up for us for today.